Hi, good evening. This is Doc Kyle, and we're gonna discuss blood. So, I think you know what blood is naman, no? It's the red thing that runs through our veins and our arteries. No, so blood in itself is just um, blood is a mixture, manggod. So if you ask me what most resembles blood, think of kanang kanang milk tea na daghang boba, no? Because blood contains lots of cellular elements, lots of minerals, lots of proteins. No, it's a very thick concoction, no? So, no, it's, it's considered as a connective tissue, no? Because what is connective tissue? Connective tissue simply means that there is what we call a ground substance, um, something that is something that connects the two cells, no? So this that it may be that these two cells produce the ground substance, as with your cartilage and your blood, no? Ay, cartilage and your bone. They produce osteoid, no? So they produce your cartilaginous material, no? So they connect each other. But with blood, the cells are suspended in the ground substance, no, which is your plasma. Plasma is a fluid component of your uncoagulated, remember, uncoagulated blood, no. So the plasma contains the liquid portion of blood, no. So because these blood cells, they can't move around, so they need to ride on something. So they ride on this plasma. So your plasma contains your electrolytes, your sodium, potassium, your proteins, your antibodies, no? So the most common plasma protein is your albumin. No? Number one, the purpose of your albumin is number one, to maintain plasma oncotic pressure. What is plasma oncotic pressure? Given that the plasma is basically water, basically water, water tends to leak if there are tubes, and then within the blood vessel there is pressure. So this pressure tends to push water outside the blood vessel. So in order to counteract that, the purpose of albumin is to have an oncotic pressure, meaning it attracts water to go in, to stay inside the blood vessel. No? So another thing, the purpose of your albumin, it's a reservoir for amino acids, especially if there is protein deficit in the body. So if, for example, you're starving, no, so you need to have protein, and then your muscles are wasted na. So they might put into albumin. That's why malnourished children usually are puffy, no? Edimatos ilahang mga nawong. That's because their albumin is very low na. They don't have any protein in their blood to keep the water in their blood vessels. That's why the water goes out to the tissues mo, mahupong siya. Oh, hupong ang kamot, di ba? Ano man ang mga malnourished, di ba? Butod ug tiyan, nana, di ba? That's just fluid that's extravasating. So we have your alpha, beta globins. This alpha and beta naming is based on your migration in your electrophoresis. Alpha being the fastest, and of course gamma being the slowest. Now your gamma globulins are your antibodies produced by your plasma cells. No, so um, all all plasma proteins are produced by the liver. Remember that all plasma proteins are produced by the liver except your gamma except the gamma globulins, because they are produced by your plasma cells, and your von Willebrand factor, no, which is produced by your endothelial cells. Endothelial cells are the ones that line the blood vessels, no. So the von Willebrand factor is important in clotting. That's why, of course, ang purpose niya kay for clotting, mas ideal if it is produced near the site which it is used more frequently. So it also contains your dissolved gases that dictate your pH, no, if you're acidotic or alkalotic. So next. So, so the cellular components, also named as the formed elements, are your blood cells. No? So, oh, yeah, so you have your red blood cells, the most abundant cell in your circulation. They are the ones, the red blood cells in itself actually are useless. No, they don't function much. The thing that makes the red blood cells significant is the hemoglobin within it. And then the thing, because hemoglobin is the active component that binds oxygen. It is the oxygen binding component of your red blood cell, no? The hemoglobin. Hemoglobin is made up of two parts, your heme and your globin. The globin is a protein component of your hemoglobin. And then your heme is the protoporphyrin, and protoporphyrin 9 plus your iron, it equals heme. And then it's where the actual binding of oxygen really is. But you need to have the globin component for it to stabilize, no? So hemoglobin is a very complex molecule. So it's the one that we actually calculate or we assess if you have anemia. It's not the red blood cell count. It's not the hematocrit, no, which we will talk about later. The hematocrit simply means if you centrifuge the blood in a tube, it's the percent of packed RBCs, no? And then maglutaw man ng plasma sa taas. So, it's not the hematocrit, it's the hemoglobin, which is more important because it is the actual oxygen binding component of the cell, no? Then next we have our white blood cells, which the purpose of these white blood cells, no, is for the purpose of these white blood cells is for the detection and elimination of any pathogen, no. It's either it's a pathogen or a foreign body. They are the ones in charge of detecting it, stimulating inflammation, and then they are the ones who eventually remember it so that we have immunity against it. So we have our neutrophils, no, which is the most abundant white blood cell. It is a granulocyte, meaning it has granules. These granules contain the chemicals that give it the function. No, when you phagocyte, when the neutrophils kill by phagocytosis. When they eat it, the granules empty their components within the phagolysosome, and then hopefully the pathogen dies. Now we have our eosinophil, the next granulocyte. The eosinophil is somewhat mysterious. Not necessarily mysterious. We have alluded that the eosinophil increases during parasitic infection. It's because the granules inside the eosinophil contain a major basic peptide. No, the major basic peptide is of course rich in your alkaline, basic good alkaline na mga amino acids such as your arginine, lysine. No? So it basta arginine na siya arginine. Hindi ko sure atong lysine. But it contains lots of alkaline amino acids, and this major basic peptide is toxic to parasitic ova. They try their best to attack the mature ova, but it's very ineffective, you know. So, and then these eosinophils also have a what we call a histaminase, no. So if you put a compound and then you put a name, no, example histamine, it is produced by your basophil again to induce allergy, no, kanang mga allergic symptoms, histamine, anaphylactic symptoms pod. So if you put an enzyme ACE, again, butangan mo na siya ACE, meaning enzyme na siya. And for most of the time, it is against that, it acts on that compound. So if the eosinophils has this histaminase, meaning it tries to cleave histamine or modulate its action, the eosinophils have a more immunomodulatory effect, meaning they try to lower the immune response to modulate it. That's why they are seen in the respiratory tract of asthmatics, no? And then, okay, they are trying to limit the action of the mast cells, no, and basophils, because what happens when basophils and mast cells degranulate, manggod, most of the time they die. So wala na tayo evidence of basophils and mast cells after they degranulate in histologic biopsies, ay in histologic specimen, no. So what we see are eosinophils who are trying their best to limit the action, no, to modulate the action of your basophils. So, um, one good thing to note about these eosinophils, no, you cannot see them in the stool. Ay makita ko sila usahay in the stool of kanang heavy parasitic load na eye burden ng mga patient. But what you see are your charcoal-laden crystals, no? These charcoal-laden crystals are the only crystals that can be seen in your poop, no? And in your sputum and in your lungs. Because these charcoal-laden crystals is the crystallization of your major basic peptide, no? So, katuha. So, not necessarily na kay parasite sa imuhang lungs, kaya di ba, nag-ato man ang eosinophil sa imuhang lungs to modulate the anaphylactic response, ay the allergic response sa asthma. But inherently, they just carry it, and then this major basic peptide, when eosinophils die, it crystallizes, no, and then that's your charcoal-laden crystals. And then in your basophils naman, your basophils are just like they're very important in a sense that they can be a killer. They can kill you in the cases of anaphylaxis, no. So always remember anaphylaxis, type 1 hypersensitivity, no. Anaphylaxis is the cross-linking of 2 IgE, immunoglobulin epsilon IgE, on the FCR, no, so fragment of crystallization receptor epsilon of your basophils or mast cells, leading to a degranulation or release of histamine, no, lots of histamine, no. So so anaphylaxis in a sense is any type of allergic reaction na multi-organ na. So we have the effects of anaphylaxis are more of a parasympathetic, no, if you remember your anatomy. So your parasympathetic responses are your increased secretions of your airways, bronchoconstriction, no, bronchoconstriction, tapos kalibanga, no. So eventually vasodilation, so that may cause shock, no, so that we have anaphylactic shock leading caused by increased vasodilation, poor na tapos kung poor na imong preload sa imong heart, lisod na imong heart, so pwede ka ma-deads, no. And then we have these lymphocytes. The lymphocytes are part of your adaptive, the branch of the adaptive immunity, no, branch of your adaptive immunity. They are the ones that remember the enemies. They remember the enemies, and then they have this memory B or T cells. So they are divided with your B, no, so B or bone marrow na lymphocytes, and your T thymus lymphocytes, no. So they are both produced, your bone marrow and your thymus your T cells are produced in the bone marrow, no, with your lymphoid progenitor cells, but they mature in the kumbi bone marrow. They mature in the bone marrow. In a sense na mature, they gain their functions there. And T, sa thymus sila nag-gain of function. No, so the T cells, they migrate to the premature thymus T cells. Okay, premature. They migrate to the thymus, and that's where they mature, no. So we also have your B cells are the ones that is your in charge of your humoral, meaning humor is a soluble na component, meaning humor, and a humor is a secretion of the body. Imong imong laway, imong mata, imong dugo, imong tahi, no, imong mga bile, no? We have 5 humors, no? By Hippocrates ba to? Anyway, going back, they are the ones in charge of the humoral immunity, no? They are, in a sense, na dili sila naa dito sa imong laway, ang B cells. They produce antibodies, no? And then these antibodies, your gamma globulins, they are the ones that are dissolved in your humors, no? That's where your protection is. And while your T cells are in charge of your cellular immunity, meaning cell to cell, naadjud siya. Naadjud ang T cell when there's a pathogen, naadjud siya, wala siya antibodies to attack that pathogen. The T cell really is at the forefront, but it doesn't kill, no? So we have different types of T cells. We have our T helper, no? And we also have our T cytotoxic, no? CD. So kato. So napa na siya mga CD4, CD8. So kato, remember, and we also have our platelets. Our platelets are same with the RBC, which is a nucleate, but these platelets are more of a fragment, mangguni ni siya, of the cytoplasm of the big boy megakaryocyte in the bone marrow. So these platelets tend to activate. They become these hairy rambutans over here, no? They tend to become these hairy rambutans. Oh, wait. These hairy rambutans over here. So why do they do like that, no? Why do they become hairy? So platelets, when they are inactivated and then there is a damage in the blood vessel, no? Kung na ay kanang there is a time na naibangag, naibuslot, if it is still smooth, then it won't adhere to the break. So it needs to have plenty projections, no? So that it will adhere better. And through these projections, it will secrete your coagulation factors, calcium, no? So it will aid in the formation of the platelet plug, which will eventually be, that's your primary hemostasis, no? Your platelet plug. And eventually, a stable fibrin clot will be done, which is the end of your secondary hemostasis. Anyway, I digress. So next is, wait, naman recording niya akong PowerPoint, no? So the plasma is the intravascular fluid which contains your fibrinogen. If you can see this 5-O-gen na apelyido, that means it's still inactive. No? So the active form of your fibrinogen is your fibrin. And this fibrinogen is it's roaming around the blood. It's quite abundant, but not as abundant as your albumin. It's roaming around in blood looking for kanang platelets to activate. Not necessarily platelets to activate it. Looking for any damage, no? So that it will plug. This is like a glue, mong good fibrin. It's like the glue that holds the platelet plug together. No? So in vitro, meaning out of the body, coagulation occurs and is stimulated by fibrinogen converted into fibrin. And then, so if there is already fibrinogen poor just means na na-convert na ang fibrinogen into fibrin. The liquid is now called the serum, no? Serum na siya. So this is a blood clot, no? Na-clot ni siya. So why there are tests, manggod, that it's better to use serum versus plasma. Example, your chemistry test, no? Prefer use serum, but of course pwede gud gihapon ang plasma, but just prefer to use serum. So because for the most part mas dako man gud ang the fluid serum. For the most part, if you let the blood clot, the volume of serum is quite higher versus plasma. And apart from that, if you use plasma and then matandog lang ni siya gamay, some RBCs might contaminate the plasma, and then when you get when you get samples from the plasma, especially in chem, you might get some RBCs which will make the results erroneous, no? So plasma has 3 components, no? So the plasma itself, the fluid itself, the yellow, and then there's also WBCs and platelets, no? It is your buffy coat. The white part is your buffy coat, no? That's where your WBCs and platelets are. And here is your RBCs, no? This is your hematocrit, no? If you centrifuge non-coagulated blood, no? So you have non-coagulated blood or anticoagulated blood, you use EDTA, ethylene diamine tetraacetic acid, no? It's a famous anticoagulant, the lavender top, no? So when you use it, what will happen is it won't make a blood clot and then mag-separate ang imuhang WBC and platelets with a buffy coat, and your RBCs will be compressed together. That's your hematocrit, no? So katuha, serum is equals to plasma minus clotting factors, namely your fibrinogen, no? So this is your blood clot because your fibrinogen is converted into fibrin. So where is the fibrin now? The fibrin is in your blood clot na. Diba? Clotting factors. Nahimo na siyang clot sila tanan. Hope you understood that. So here, kato lang. Gani siya ang components sa imuhang plasma, no? So we have your protein, your globulins. Globulins are just in general, globulins are just proteins na nag-form of globule. Wala siya, no? So, you have your proteins, albumin, fibrinogen. It's just electrolytes, nutrients, and respiratory gases. Respiratory gases are the ones that dictate, for the most part, they dictate your pH of blood, no? Stop talking. So here, RBC is the most abundant cell, no? So they are enucleate, meaning wala sila nucleus. And their purpose is to carry hemoglobin. So like I said earlier, compacted RBCs, your hematocrit, is about 45% of all blood volume. About 7.5. This depends on the source, no? 7.5 ni akong book right now, but there are some that uses 4 to 6 no? But the most the most reliable, well, not necessarily reliable, this is the most apparent this is the most commonly used comparison to the RBC to say if it's a normal size is you compare it with the nucleus of a small lymphocyte, no? So this is a lymphocyte, no? It's just one big nucleus and a very scant cytoplasm. So this is the small lymphocyte. There are different types of lymphocytes. Manama to yung mga typical giant lymphocytes, reactive lymphocytes. So this is a normal small lymphocytes. The lymphocyte has a blue-colored cytoplasm. And Robin's egg blue ang iyahang description in some books. So this is a red blood cell. It's biconcave. There is what you call a central pallor. In a normally, the size of the central pallor is usually less than 1/3 of the entire size of the cell. You know, because if it's going back to my previous explanation, I think na-explain naman ako ni sa bone marrow na topic. But the red blood cell is pink because it has a lot of hemoglobin. And this hemoglobin has a globin component which is usually an alkaline na component. So eosin, the stain that we use, usually binds to this is an acidic stain, and then as an acidic stain, it binds to alkaline components or compounds. So that's why, and eosin is colored red. So it binds to the hemoglobin, the globin portion of your hemoglobin rather. That's why red siya. So we can quantify, if we use the PBS, we can quantify whether or not it's normal because, diba, central pallor, meaning wala kayo hemoglobin diha. If the hemoglobin is low, meaning your central pallor would be enlarged. No, like this one, it's enlarged. It's greater than 1/3. Actually, na siya grading. Mild, moderate, no, and severe hypochromia. So that's what we call it, hypochromia. No? Hypo meaning low, and chromia meaning staining, meaning hypochromia. Dili ka siya mag-stain because there is low hemoglobin. So the central pallor should be usually less than 1/3. If it's greater than, there might be a hypochromia. But like this one, it's hard to say that it's hypochromic because it's just one cell, no? So you have to look at the bigger picture pa jud. So it's, if most of the cells you see are kanang hypochromic, then maybe the patient has hypochromic anemia, no? So next, this is what we saw ganiha gyapon. This is a cute acute RBC, no? So nana siya central pallor. So apart from, like I said ganiha, in the bone marrow, apart from the explanation that it can move easier in tight spaces if it's a biconcave disc. Another thing that can be noted with the biconcave shape is it is more efficient, no? Because it is more efficient in oxygen transfer, because if it was around just a spherical, no, there is a condition as spherocytosis, spherical RBC, the hemoglobin in the middle would have a hard time transferring the oxygen because it's quite far, no? So given the shape, the biconcave shape, the hemoglobin in the middle has doesn't there is no inhibition to adequate oxygen transfer because of course mas nipis man ang sa tunga, no? So kato, so here what we see is a rouleau formation. Ideally, no, in kanang medium sized vessels, our RBCs don't dikit jud, no? They don't dikit, they don't overlap rather, no, in the circulation. That's because we have what we call our zeta potential, no? Zeta potential is a negative charge. So kung negative ang mga RBCs nimo, 'di ba? Opposites repel each other. If they have a mild negative charge, meaning they will repel each other. That's the zeta potential. It's brought about the sialic acid. Oh, sialic acid ata to sa ilahang membrane. That's the zeta potential. But here, in your small blood vessels, and since they don't have no place to go, mag-stack yun na sila eventually due to the pressure. The pressure will eventually override the negative charge, no? So, kato, they will form a rouleau, meaning a rouleau is just a stack of coins. No, that's the origin of the name rouleau. And they will stack together. So this principle is what we use in your erythrocyte sedimentation rate na test. Your ESR, basta, it's the speed, no, the rate, speed on how fast mag-stack imuhang mga RBCs based on the increase of your acute phase reactants. Anywho, wait siya. So RBCs have a biconcave shape. So RBCs have a biconcave shape to increase surface area, no, like I said earlier, and to help conform the blood vessels to the sinusoids and facilitate blood gas exchange. Always remember that hemoglobin is the one that facilitates oxygen carbon dioxide transfer. Hemoglobin consists of protoporphyrin 9 and iron, alpha and beta chains. So kato, no? So the usual hemoglobin is your oxyhemoglobin, which is hemoglobin with oxygen bound to it. That's your oxyhemoglobin. It's bright red in color, no? That can be seen in your arteries, no? And then your carbamino hemoglobin, wait lang, can't see it. Your carbamino hemoglobin is your hemoglobin with um, CO2 bound to it, no? Na-exchange na siya. This is usually seen in your in your veins. And then carboxyhemoglobin is with your carbon monoxide. It's cherry red, no? Remember ha, carboxyhemoglobin isn't carbon dioxide. No, so it's carbamino. And cherry red ang imuhang carboxyhemoglobin. Next. So here, this is what I said ganiha, is red blood cell. So this is your beta chain. And then your alpha chain. So it has two chains, two alpha chains and two beta chains. This is the adult hemoglobin, no? So we have our fetal na mga hemoglobins, no? Your goer 1, goer 2, no? We have your epsilon na anya. So kato, epsilon, gamma man ata to na chain. So this spaghetti is your globin portion, no? And then this sunny side up is your heme group, no? So this is protoporphyrin 9, kaning yellow, and then your iron is in the middle, which it binds to the oxygen, no? Sige, next. So your white blood cells, no? They act as the... Shut up, me. No? So they are security system, no? So they from both external and internal threats, no? Your external meaning your mga pathogens, katong natunog kag lansang, no? Katong Bacillus anthracis na naa dito, no? Uh, Clostridium tetani na natusok kag lansang, no? And then your internal threats are your cancer, no? Your malignant cells. So generally inactive within the blood, no? Like I said earlier, granulocytes and granulocytes, so that they don't damage. So when you activate manggod your white blood cells, wala man gud sila dagan kay collateral damage ana, no? Unsa na sila, they don't mind killing the surrounding tissue. That's why when you have an infection, sometimes the endpoint is mas dako pag damage. The damage produced by your WBCs far exceeds the damage that the pathogen made, no? Same with your hepatitis, no? The hepatitis B virus, it doesn't, negligible lang iyahang damage to the hepatocytes. There is, but the main the main damage, the main cause of damage in your liver with your hepatitis virus, no, hepatitis B virus, is the immune system. So transmigration about outside the blood vessel is occurs via diapedesis, no? That's from your neutrophil. So your lymphocytes migrate via enteropoiesis. Well, for the most part, no, they can also diapedesis man po. Diapedesis is a general term of moving outside the blood vessel. It's usually done by your neutrophils. No, so how do they diaped? The diapedesis, that's why your neutrophils on your granulocytes, no, so they really migrate, monggo, they really do diapedesis. That's why they have a lobulated na nucleus. Because if they need their nucleus, monggo, they need their nucleus because it holds abundant genetic information to produce proteins. But they can't lab around a solid na nucleus. Di man sila makaagi sa mga, they can't pass through the pores of the blood vessels, or they'll have a hard time. So in order to litigate that, to work around that, God designed them, the neutrophils, to have a lobulated na nucleus so that it will be easier to pass, no? Their overall width is smaller but their overall volume is the same. So kato, no? So we have our neutrophils here, what we call azurophilic, neutral staining, meaning it's very light. Actually kanina klaro na ning gwapo ning camera ani, because you really can see the granules of the neutrophils, but generally it's really hard no, to see because it's very light, lavender, lilac na mga granules pag neutrophils. If the granules of the neutrophils become more coarse, that's what we call toxic granulation, especially if the infection is really, it's a really heavy infection, no? So kato, neutrophils are the first ones to migrate to the area, no? Because not only are they the most abundant granulocyte, they are also the ones most active, and they are the well equipped in general, no, to attack pathogens, no. So they have an all-around killing power. That's because they have their myeloperoxidase, no? So next is our eosinophils. So from the name itself, eosinophil, eos meaning red. Actually, it means the dawn. Eos meaning dawn. Dawn ba? Kadlawon. May mga red na pula-pula na kadlawon, di ba? No? So, it's red. So, it has lots of major basic peptide, no? In its granules. Diba? Because eosin is an acidic stain and it binds to alkaline components. While your basophil is a basic staining, meaning blue basic. So, if it's a basic stain, meaning its granules has acid. So, it has lots of acid. Your heparan sulfate, mana, acidic man na sila. No, it has lots of acidic components inside its granules. I'm not sure with histamine if acidic bang histamine. But kanang mga heparan, more or less acidic na sila. That's why it stains blue, because it has acidic components. You know, so your lymphocyte is this one, scant, gamay lang iyahang cytoplasm. Cytoplasm is scant, and there is a well circumscribed dark staining nucleus. That's the difference between your lymphocytes and your this is most probably a T lymphocyte. No, it's not a B lymphocyte, because the B lymphocyte has a bigger cytoplasm. It needs a bigger cytoplasm and a perinuclear Hoffman sa B lymphocyte. So the main way on how lymphocytes kill is through injection of cytotoxin, no? Sa mga cytotoxic T helper. So, cytotoxic lymphocytes there rather. So this is your monocytes, no? So remember from my bone marrow lecture that they are, even though they are separated by having granules and no granules, they are part of your myeloid lineage, no? This is your monocyte. The nucleus of the monocyte is kinked, no? Mura siya kidney bean, no? Kidney bean. Coffee bean di ay. Mura siya kidney o coffee bean. No, not siya granules, but like I said, this camera that they use, no, is very beautiful because you can see the granules. But for the most part now, everyday na to na paglantaw, it's really hard to see the granules of the monocytes and the neutrophils, no? So the monocytes are the circulating population, and then when they reach the tissue, they become macrophages, no? And pag mahimo na silang macrophages, o no holds barred na, so sige lang sila kaon, no? So this is the neutrophils, no? So faint, light pink or lavender, lilac, mga alam mo ng mga unsa niya. Faint lang jud ang iyang granules, no? 3 to 5 lobes your neutrophils, no? In some books na pag mo greater than 5, hypersegmented na siya. Your eosinophils is bilobe, no? Red, dark pink. Gamay ra kay na siya. Actually, pag makakita kag 5, um, it's very common to see eosinophilia, especially because there are lots of undiagnosed asthmatic patients, no? So here, kill helminthic. Helminth is your parasites, no? And modulate local inflammation. So next is your basophils, dark blue or bilobe. So modulate inflammation, release histamine during allergy, and then your granulocytes, your lymphocytes, no? So the most common is your neutrophils, and then the second most common is your lymphocytes, no? Irregardless na kung B or T, no? So we need flow cytometer to know if it's B or T, but for the most part, lymphocytes ang second. So they are effectors and regulatory cells for active immunity. And then your monocytes, no? So the usual most common ani is your neutrophils, lymphocytes, monocytes, AO, and basophils, no? So it's very rare to see a basophil, but in cases of anaphylaxis, you can see it in the PBS, no? You can listen to my recordings man if naboringan mo sa video. No, so granulocytes, neutrophils, and eosinophils. I won't take this off the PowerPoint, the recording. No, so here, neutrophils and eosinophils are capable of phagocytosis, but all have specialized lysosomes called azurophilic granules and smaller granules. No? So your granules are just lysosomes. Your lysosomes bind to the phagolysosome. No? So kato. So your neutrophils and eosinophils are capable of phagocytosis. Ha? Walay labot ang imuhang basophils. Here, oh. Kidney shape ang imuhang monocyte or your kidney shape or coffee bean. So it needs to reach the site of the infection pa, no? To enlarge and differentiate into macrophage. Usually in the bloodstream, in the blood vessel, your monocyte is inept, no? Wala na sila ginabuhat kayo. So remember your neutrophils 3 to 5 ang lobes, eosinophils and baso usually bilobe na siya. Your basophil is single lobe na S. So the granulocytes, going back to the neutrophils, are polymorphic, no? Multiple morphologic orientations. Okay, they're very fluid, magud your neutrophils, no? So they can change whatever they want their morphology to be just to get to the point, no, and eat those bacteria. They're multilobulated nuclei, so enable them to squeeze into the spaces. Neutral staining, lavender, lilac, not very blue, but not very red, no? So, usually, lisod lagi siya tanaw, makitaan sa microscope. So, the cytoplasmic granules of the neutrophils provides functional activities and are two main types. Your primary granules, or your lysosomes, no? Azurophilic, azure, meaning bluish, azurophilic, no? So are large, dense vesicles, because they contain your myeloperoxidase. So this is the enzyme that produces your hypochlorite, also known as your bleach. No? Ano yung sodium hypochlorite ninyo? Bleach na. So, gahi kaya na siya mapatay og kagaw. And it also produces your lysozyme, which degrades the bacterial cell walls. No? lysozyme is effective against your gram-negative bacilli, no? So it also has your defensins, your cysteine-rich proteins that bind and disrupt the cell membranes of many types. No. So the next page. So then we have your secondary granules. No, they're smaller, and then they're less dense, and they're pinkish. No, so they have including secretion of various ECM degrading enzymes. What is this ECM? That's your extracellular matrix. No, so example, the bacteria hid itself within the extracellular matrix of your kani sa imong connective tissue sa imong wound, no? Neutrophil don't care. Neutrophil don't have time for that. So patcho niya tong unsa, bungkago niya tong gisuksukan sa kagaw. No, that's why neutrophils are very they don't care about collateral damage. You just kill everything in sight, no? So they have this many mga hyaluronidase, no? No, so we have phosphatase, collagenase, no, hyaluronidase. No. So they also kill the cell wall here, collagenases, because some of the cell wall of the bacteria have collagen. So kato. So excuse me. Your azurophilic granules, your primary granules, so remember it has your myeloperoxidase jud ha, producing hypochlorite. Mao na siya importante, no? Then your secondary granules has your lysozyme, no? Ulo, nalagay sa secondary granules ng lysozyme. Anyway, point of contention yun lang siya. So I will not ask na lang where lysozyme is seen in the primary or secondary granules. No, so tertiary granules, no? So kato. So basta remember guys ha na the primary granule has your myeloperoxidase. Mao nang importante ha. Kanang jud. Has 3 populations, but dito sa akong bone marrow na lecture, it has 4. No, that's why I use this book, no, is what I prefer, because information really varies from one book to another book, no. So it's maybe due to politics, no, or mas prefer lang sa isa ka researcher ni na data because it's better to correlate with some conditions, no. So but we as students, I still consider myself as a student, we as students should know most, no, should know most, meaning we should know the perspective of this book and the perspective of that book, and we should conclude for ourselves which we will use, no. Ang importante, kabalo ka na nakay reference. Kaya kung ikaw nagbuot-buot lang ka, wala kay reference, that is a fallacy. No? So, has 3 populations in this. So, we have the bone marrow population. So, combine na niya tong 1 and 2 ato sa bone marrow. So, it's back up. And then the marginal population, these are the ones that I told you that they are always attached to the endothelial lining. No, so they are the police on the outposts, no, and the circulating population doing the rounds in the vasculature, no, the ones represented in the CBC. So kung magsuka, moapil kanin sila na ang duha na makitan, no, because the marginal population will mobilize. So your eosinophils. Your eosinophils are bilobe, no, less numerous than your neutrophils, and they have large acidophilic, no, acidophilic ilahang granules because they have lots of alkaline components, no. Specific granules, typically staining pink or red. Acidophilia is due to the abundance of your major basic peptide, large concentrations of your basic amino acids such as arginine. Granules also contain histaminase, no? So, na-explain naman ako ni Dinhe. No, it's parasitic, toxic to parasitic infection ng MVP. And then the crystals seen in stool is your charcot laden. And then the eosinophils are active in immunomodulation because they destroy the histamine with their histaminases. So this is your eosinophil, no? Eosinophil ba ni? Ano? Marble ceremony siya? Ayun, yeah. This is your eosinophil because it has MVP. No? So, it's a long time. So, kung siya daghan siya mga pakapin. Next, it has sombrero vesicles. Next is your basophils. Your basophils are less than 1% of the circulating leukocytes. No? Leukocytes meaning your white blood cell. No? Their specific granules stain purple, the basic dye, meaning they are basophilic. So they have acidic na they have acidic na mga components within their granules, no? Presence of heparin din acidic, sorry guys. Heparin, no, and heparan sulfate, no? Heparin mo lagi. And other sulfated, what's GAGs? Glycosaminoglycans, no? So the basophilic specific granules contain much of histamine also. So including platelet activating factor. And why is this eosinophilic chemotactic factor, no? So since this because in the body there is when some process occurs, no, example here in allergy, in the if example na mobilize yung basophil and then mag-action or release siya histamine, at the same time release po niya yung eosinophilic chemotactic factor so that na ay mag-counterbalance iyang effect, especially if the effect already has done its purpose, no? So since nagpagawa siya histamine, at the same time magpagawa siya eosinophil chemotactic factor para katong eosinophil siya ang mag-modulate sa immune response, no? So basophil lang siya saba at the same time gitawag niya si eosinophil para pahilumon siya. Something like that. So associated with type 1 anaphylactic, no? So there must be cross-linking of the IgE so that magdegranulate imuhang histamine for parasympathetic stimulatory effects such as vasodilation, bronchoconstriction, and pruritus, katol-katol. So we have basophils and we have mast cells, no? They are related. They have the more or less the same function. And actually, we still don't know if they have the same progenitor cell. No. But usually, the mast cells are seen in the tissue. Usually, you can see mast cells gyapon sa blood. And then, but they have the same, more or less same the function to release histamine. No? They are still the ones that initiate your allergic response, type 1. So we have granulocytes na. So the most numerous are granulocyte in the blood, no, second only to the neutrophil. Sorry. The most numerous are granulocyte kay gamay lang mo po ng population sa imong monocyte. No? So the smallest usually it's the smallest leukocyte, the smallest white blood cell. The lymphocyte is the smallest, diba? It's just the same size of your RBC, ang nucleus. So, we can differentiate your lymphocytes using the cluster of differentiation, or CD. CD is just like your different receptors in the cells, no? So we have our, and DCD, because since lymphocytes look the same, no, lymphocytes look the same, we must use the kanang receptors to tag them, to identify them. No? So that's their cluster of differentiation. So your B cells mature in the bone marrow, like I said ganiha, humoral immunity, due to the production of your soluble immunoglobulins. That's the immunity you see in your humor. So they are most common with, and then their receptors, no, ilang ID is the CD19 and CD20. And your T cells, the one that mature in the thymus, is in charge of the cellular immunity. Kay sila man jud ang, they have direct contact with the pathogen, no? So they kill. Think of it as the T cells are in charge of the intracellular na mga pathogen, no? And not just intracellular pathogen, but mga within. They're like the internal police, no? So they're like the MP, military police, no? They are the ones policing our own cells, no? So they kill the tumor cells, no? Think of viruses as spies, manggod. Mga spy ba? They are the ones that kill the virus, good. So CD4 is our T helper cells, and our CD8 is our T cytotoxic. So your T helper cells are the ones that modulate or initiate inflammation. Your cytotoxic cells, they directly kill tumor cells, no? And virus infected cells. They have the same action, more or less, with your natural killer cells. But your natural killer cells, CD16 ni siya, your natural killer cells, hindi na din makitaan. Your natural killer cells, no? Your CD8 man god, your cytotoxic cells, sometimes they have memory. So they know you. Kani siya, nibalik na po ni siya, patso na to ni. Kani siya sa una, ni unsa man ni siya, ni kanang kagaw man ni siya sa una, so patso na to ni. While your CD16, chuy lagi ni siya, first time man ako nakita ni siya, pero chuy lagi ni siya, patso na to ni dayon. No? So they are somewhat, although they are still in your, pwede sila ma-use mo ang adaptive immunity, but innate ang meaning kong innate, there's no need to be stimulated, there's no memory kumbaga, no? So if makikita ni siya, if the natural killer cell sees something unusual, no, especially if na yung mga unusual pathogen or kanang dili normal flora na kagaw, or mga there's something wrong in the tumor cells, there is no hesitation, there is no checking sa diretso patay, no? Unlike your T cytotoxic cells, no, first magmatch sa with the memory, the memory bank, no, before nila mag-act upon it. So that's the somewhat major difference between your NK, natural killer cells, and your T cells, cytotoxic T cells. So this is your monocyte, big boy, no, kidney bean, coffee bean or kidney shape. Your lymphocyte, no. This is your platelet, your platelet. So this is a monocyte, no. So they are the precursors for your macrophages, osteoclasts, and microglia. Remember that the osteoclasts are macrophages, no. So anything that chomps, that eats, no, is macrophage, you know. So circulating monocytes have diameter, they distinctively have an indented, olay na po ni C-shape daw ang iyang nucleus, no. So here less condensed ang chromatin nila than your lymphocytes. Many lysosomal, small lysosomal, and azurophilic granules. Usually dili jud nimo makitaan. Usually it's really hard to see the granules of the monocyte, no. So sorry. So next at us, platelets. The last but not the least daw is your blood platelets or thrombocytes. Very small, non-nucleated. membrane brown cell fragments lang din siya. So, they originate from your megakaryoblast, no? Megakaryocyte. So the biggest cell, biggest hematopoietic cell in your bone marrow. So this megakaryocyte has plenty cytoplasmic projections, and these projections chip off, which will lead to become your platelets, no? So the platelets promote blood clotting. They are the ones that adhere. Example, na break sa blood vessel. They are the ones that adhere first because they have receptors for discontinuous na mga blood vessels, no? So they form a platelet plug, and at that same area they release their clotting factors, and then their clotting factors ug mga other substances that will both stimulate inflammation and clotting, no? Eventually, the secondary hemostasis will be done, forming a stable platelet, fibrin clot, no? So, next. So, these are your platelets. They're quite cute, no? Fragments lang sila. Actually, we can do a platelet estimate by using the peripheral blood smear lang. And this big boy over here, is your megakaryocyte, no? Look over here, nana kay mga platelets din na, no? And platelets man po ni. So, the megakaryocyte is big boy because it undergoes endomitosis. Endo, meaning mitosis, no? Meaning mitosis from within. Meaning instead of producing daughter cells, it just keeps everything inside, no? So, maduplicate lang iyahang nucleus din ha. It's exponential, no? So, 2, 4, 8, 16 na nucleus na siya. And the cytoplasm also gets bigger, no? So, why is that? Because it needs to produce lots of protein. Protein that will go inside, that will form its projections, and protein that will go inside the granules of the platelets. So, kato. Oh, it's done. No, so that's blood in general. If you have questions, feel free to PM me or ask me. According kay Doc Ian, atong activity kay, well, I also suggested that ay activity nato for this week is just identification of the peripheral blood smear, and hopefully makuha ninyo dali man na siya. Sa pula, eosinopil. Sa blue, basophil. Sa kidney, monocyte, no? Mao lang yun na siya. So if you have any questions, just feel free to PM me, and thank you. Bye bye guys, God bless.